Assalamualaikum and good afternoon, everyone. My name is Sheril Ankasha, and I will be presenting a study with title Mycoran A200C3P regulates metastasis by binding to 3' untranslated region of DLC1 in high-grade serous ovarian carcinoma. In Malaysia, ovarian cancer is the fourth most common cancer in women and is the leading cause of death for patients with gynecological cancer. High levels of genetic and molecular heterogeneity are reported for epithelial ovarian cancer, and these epithelial cells can be transformed into different histological subtypes, where serous ovarian cancer is the most common. So, this serous ovarian cancer is classified into two categories, namely low-grade serous carcinoma and high-grade serous carcinoma, which is HGSC. In comparison, HGSC often diagnosed at late stage, either at stage 3 or stage 4, in which the tumor growth rate is aggressive and has spread to and metastasized to omentum and mesenteric areas. The condition leads to a low patient survival rate. Therefore, extensive research and a better understanding of the molecular changes in the progression and metastatic process in HGSC are crucial in order to develop a more effective therapeutic approach to overcome this life-threatening disease. MicroRNAs are small non-coding RNA with approximately 20 to 22 nucleotides in length. They recognize and bind to partial complementary sequence of the messenger RNA, commonly at 3' untranslated region or 3' UTR. This could resulting in gene silencing either by translational inhibition or mRNA degradation. By this mechanism, microRNA can play an essential role in biological process by negatively regulating gene expression of its targeted gene in normal and pathological cellular process. In cancer, it can act as either oncogenic, tumor suppressive, or metastatic microRNAs based on their functional activities. Growing evidence has indicated that the unique expression profiles for various cancer types could help in identifying tumor-specific microRNAs and their targeted gene for the development of novel molecular diagnostic and prognostic markers, especially in HGSC. In Malaysia, for the first time, we have identified a list of up and down regulated microRNAs in serous ovarian carcinoma in our previous microRNA profiling study in the year 2015 and 2020. And we have found that microRNA 200C was significantly upregulated. In the in silico analysis, by using several databases and algorithms, we also found that deleted in liver cancer 1 or DLC1 is the target gene of microRNA 200C3P. So in this current study, we decided to continue researching the role of microRNA 200C3P regarding its targeted gene DLC1 and try to elucidate the underlying molecular mechanisms involved in the progression and metastasis of HGSC. As the methodology, an oligonucleotides containing the binding area of microRNA 200C3P were cloned into a vector and transiently co-transfected with microRNA 200C3P mimic in SCOV3 cell line. However, in order to get the predicted binding region, two web-based services recently developed were used, which is TargetScan and microRNA.org. As the result, based on the luciferase assay, the first figure indicated that the luciferase activity reduced by 80% when the full 3' UTR of DLC1 construct was co-transfected with MIR200C3P mimic. From the in silico analysis from the full sequence of 3' UTR of DLC1, there are two putative binding sites predicted by bioinformatic prediction tools. And to identify the specific binding area of microRNA 200C on 3' UTR of DLC1, two firefly luciferase reporter vectors containing site A or site B, namely as wild type A or wild type B respectively, were constructed. This figure shows that overexpression of MIR200C3P significantly reduced the luciferase activity in wild type A by 44%, but not in wild type B. This showed that the binding area of microRNA 200C3P could be within the wild type A region. We further investigated whether these two binding sites were repressed by microRNA 200C3P. So three additional luciferase vectors were constructed containing the mutant DLC1 3' UTR sequence with deletion for both binding sites, which is mutant A and B, and either of the two binding sites, which is mutant A or mutant B, as you can see at the figure on your right. As shown in figure on the left, mutant B showed a significant reduction in luciferase activity, whereas there is no significant reduction in mutant A and mutant AB with the presence of microRNA A200C3P. So these results confirm that microRNA A200C3P repressed the DLC1 gene through its binding in the 3' UTR at site A. To confirm more on the specific target binding site of microRNA A200C3P on 3' UTR, a small region containing site A and site B was isolated into different vectors, namely as wild type A with 46 base pairs and wild type B with 53 base pairs. Additionally, two vectors containing 3 substitution mutations on the first 3 nucleotides of each seed sequences were constructed. As you can see on the left figure, with the presence of microRNA A200C3P, luciferase activity was significantly reduced by 25% in wild type A and 20% in wild type B, but not in both mutant type containing vectors. So in this study, we clearly proved that with the two putative binding sites predicted from the bioinformatic analysis, we have verified and confirmed the interaction between microRNA A200C and 3' UTR of DLC1 in luciferase reported experiments in SKOV3 cell line. As a discussion and conclusion, an understanding of the interaction between microRNA and mRNA and the validation of the targeted site involved could potentially assist in microRNA-based drug development in the future. Therefore, we suggest that a combination of microRNA 200C3P and the molecular target sites of DLC1 could be a potential regulator in high-grade serous carcinoma for therapeutic purposes and to reverse cancer progression in this deadly disease. That's all from me, and I thank you.